deck, Hercules is past the transom.
Roger.
Got all sorts of twisty turnies in there. There we go.
Okay, awesome.
Nav, deck, we have tether all the way out.
Roger that, tether out.
Data, did you copy that?
I did. I didn't have you guys on to listen to you, so I heard it now. Thank you.
CTD.
Do you have tether wraps zeroed?
I do not. Yeah. Now I do.
Awesome.
Thank you for the reminder. That's so helpful. That's a great addition to your list.
Yeah.
Yeah. I don't know how we didn't do that years ago. Turn some lights on.
Nav, can you have Hercules pull forward, please?
Fine.
Roger, pulling forward.
Definitely a good cross current. That's fine.
Nav, deck, can you have Hercules do normal operations for the rest of the launch?
Roger.
Okay, this marks the start of the vertical video transect. Hopefully data copies that.
Data copies. Thank you.
Nav, deck, Atalanta's away.
Atalanta's away, roger.
Oop, not that button, there we go. And we're gonna start doing this.
Trevor, were you in the van last time when we...
Yes, I am, yeah.
Hercules, dive.
What was that?
Trevor?
Roger.
Stand by, Fabio.
Okay.
Who said that? Okay. Roger. Hello, Fabio.
Hey, how's it going? Good?
Good.
No, just about the lighting setup. That I'm not sure if you were here yesterday.
Yes,
that we're
it's the same.
turning off the down lights.
Yes.
Great.
Fabio, what are we looking for in this transect?
We're looking for...
Can I keep comms to a minimum just until 50 meters, please?
Sorry.
Thank you. Ground faults are still good. I did see a low one on craft, but I think we can move that on descent. Yep, perfect. And what is that camera normally? Yeah, roger that. Thank you. I don't know what that one is. I have 1, 2, 3, 4. You have 2. It's not an ROV cam.
Speak. Hey, video. Can we just step the zoom a little bit after? I think there's still a bit of vignette effect there.
Oh, okay. On Zeus.
Yeah, go ahead. Go ahead, video.
Thank you.
Did that fix it?
It looks
Thanks.
it looks
Or
good.
what you're talking about? Or are you talking about Argus?
Okay, 32 meters, 9.8 meters a minute. Speed up a little bit. Ground fault's good, gauge is good. What's that? Okay. Nice. How do I see the, here we go, 10 meters. All right. .41 meters, almost at handover. It's a lot slower with the vertical video transect at this first 50 meters. Drags out the pain. I just want to get through these 50. Good, good. How do I see over here? Yeah. Okay. I'm going to carry on through the 50 meters to maintain the video transect, so when you get handover.
Nav, deck, all stop at 50 meters. Control has been sent.
Roger, 50 meters.
Okay, sounds good. I'm actually behind you anyway, so that's perfect. And, yeah, okay.
Looks like there's more light than we're used to. Is that something different there, Trevor?
Just this daytime. I think last time we launched at night. You
We're
can double check on my hotel page. I'll show you the lights. We have upper, mid, starboard port, and aft on. Only down and scale laser are
okay.
switched
Yeah,
off.
yeah. 50 meter. We still have a lot of daylight going on. Yes. Wow.
Whoa.
Look at that.
Sure, you can hold position. Beautiful.
Jesus.
Ooh, 11 meters. Neat.
Wow, look at those tentacles.
Okay. Hi, SPL. We can do normal SPL conversation. Everything looking good on the vehicles, and launch proceedings are complete. Just going down.
Sorry about that, Trevor. Thanks
That's
for the
all good.
reminder. Got
Thank you
so
for your
excited. Maybe we can just kick it off with the usual. Who is in the van right now? Maybe we'll do the back row first. Let front row get settled. So, welcome everybody. Of course, I am Monica, and happy to be joining you. We are beginning our dive to part of Endeavour known as High Rise, and we're going to check that out in a bit. And to my immediate right, I have the wonderful and talented
Fabio Delel. I'm a senior scientist with Ocean Networks Canada, specialized on deep sea environments and biodiversity. And to my right, there is the amazing
I
dive
thought you were referring to Cassie.
dive chief. Dive chief.
I'll be leading this dive today. Derek, also
What
with
happened to
ONC as well.
You fired Dougie already?
Next to me is Cassie.
Yes, hello. I am Cassie. I am also with Ocean Networks Canada. I am sitting in the data logger seat today.
Wonderful. And maybe I'll pass it over to Nav if you're ready.
Hey, this is Lynette in the nav seat.
Great.
This is Trevor in the Herc seat.
Oh, Gabby, you're not on SPL.
This is Gabby in the co-pilot seat.
Lovely. And video, who are you?
This is Ashley with OAT in the video seat. Super excited about this dive.
Fantastic. Why is that?
I don't know, I just have a good feeling.
Awesome, possum. And Fabio, maybe back to the question. You are doing a visual transect, and you've asked for reduced lights. Why is that?
Oh, look at that fish there, Fabio.
Yeah, that's...
Well, that was a non-normal fish, eh? Did you see that?
I don't know what that is. Just this shadow. Yeah, it's just to reduce... we're trying to minimize the glare in front of the... right at the field of view in the camera. And I think we made some experiments in the previous transects.
Sorry, Fabio.
No worries.
Mm-hmm. Good call.
I didn't mean to interrupt. Go ahead. I should have just gotten off SPL.
And then this downlight that we kind of removed it, and then the illumination field is much better now, so we can see the zooplankton and gelatinous plankton.
Speaking of, there goes a little tiny copepod. Whee!
Not so tiny, it's a giant.
Is that true to life or is that magnified?
Is what?
Is that magnified or is that true to life?
Well, it's close to the lens of the Zeus Plus, so it's definitely magnified.
Cool.
But it's
See what that
normally
does.
it's very hard to see copepods.
Okay.
Oh, look at that. That's a cool shot.
Mm-hmm. So for those who are looking, that little T that's floating around is a copepod.
Are you going to be able to do that while keeping it off to starboard? Use 180
Yeah.
cam and like...
I'm not going to move it. I'm just going to turn it on and off.
Yeah, roger.
And some folks wondering if we've got Argus or Atalanta with us today, and it is
Yeah, that's
Atalanta.
It's just to,
Yep.
Argus is at home resting.
just to open the valve.
Just to totally anthropomorphize them, at home resting. Now, Fabio, how deep is this transect expected to go?
To 1,000 meters. Our station is close to 2,300, right?
Yeah.
But we're gonna just do the top 1,000 meters of the water column. Hey, video, it looks like the image is a bit out of focus. Can we take a look at that? Yeah, looks like it got better there. Definitely improved, thanks.
And what's our descent speed for this?
10 meters per minute.
10 meters per minute. Very good.
Where does that number come from, Fabio?
Well, that's kind of a threshold where we can see organisms without, causing turbulence, and they actually, we can image them and have the time to identify them.
Right. Cool.
There's a bunch of some misses, I guess.
I can.
I'm not gonna listen to blue.
Oh, I'm not going to listen to blue either, so whatever. Unless you're playing country.
I'm just looking at the dive procedure here. Have we verified that the 2 porch beacons are responding?
So welcome for those who are just joining us. We are starting our dive and we are doing a visual survey. So some of our lights are off, which is why you'll notice that some of the feeds are a little bit darker. And this is to disturb the wildlife a little bit less. Our pace as well is meant to also be a little bit less disruptive to the wildlife. Herc, although he's awesome, he's a little bit of a Hummer on safari. So if we want to see the wildlife, we got to turn the tunes down, got to turn the lights down, make sure that we're not going to spook anything. And so part of our descent is to watch for the crunchies and the squishies that go by.
And just to confirm to Dirk, the dive lead here, yes, the ONC CTD is on and streaming data.
All right, thank you. Yeah, I mean, anything's fine. Don't listen to anything on my account. I'm happy with anything. Or I can just turn it off.
Did you hear that, Data?
Yes, thank you.
What was that? The beacons are pinging? I didn't hear that.
Yeah, we are not getting a position for them, but they're also on the front of the Herc porch, so we have like the whole Herc vehicle between the beacons and us.
Yeah.
And they're also, yeah, on their side.
Yeah, both those beacons we did test prior to the dive, so
Did you dangle
they
them on
should
a
be
line?
working.
Dirk's not listening to me.
What's the question?
Did you dangle them on a line off the side of the ship?
Yeah.
Hang 'em? Yeah.
Yeah.
Okay.
What's the plan with the beacon, Dirk? Beacons.
Oh, so towards the end of this dive, we're going to recover one of our RC moorings. It's a regional circulation mooring for studying the plumes. And it's about a 250 meter tall mooring. But the last step of the dive is recovering that one, and we'll replace it with a new mooring tomorrow. So what we do is we hook a beacon onto the top of that mooring so that we can track it on the way up. Just so we can know, we can kind of expect where to find it or when it's going to hit the surface.
Is it likely that it will, pardon the term, wiggle on its way up, or is there enough flotation that it'll travel pretty straight?
No, it'll travel
What's
straight.
this?
I think it's about
Is that
600
Atlanta?
pounds of buoyancy on the float. So
Cool.
probably expect it to cover the 2.2 kilometers in about, I don't know, 20 minutes.
Cool. Do you think it'll porpoise right out of the water? Like,
No,
No?
they don't breach.
oh, okay.
We'll just,
Darn.
you'll see it. It makes a good, I don't know. It's something.
It's
but
something.
it's not like a missile.
Oh,
And the other beacon, before recovering the ROV at that same spot, we'll put a beacon on the bottom so that we know when we're deploying the new one, we'll deploy it using the ship's winch wire. So we can place it in exactly the same spot, and that way we know our cables will reach. Because all of these moorings are, well, most of them anyways, are plugged into the network. Hard part is you got to put them where, so your cables aren't too short.
right. So these are cabled moorings, not autonomous moorings.
These are cabled moorings, yeah.
Cool.
So what's the consequences if these porch beacons are actually not talking?
The consequences, I would say not that, it's not, there's always something we can do.
Okay.
Tracking the mooring on the way up is not critical, so we've recovered them without doing that in the past.
Roger.
If neither works and we can't mark a location on the seafloor, then what we could do is probably, I don't know, see if there's a safe landing zone that's kind of conservative for the cable to lower the next one down anyways.
Right.
Or we could send a new beacon down like we did yesterday on the node dive and move it into the right location as a mark while Herc's still at the bottom.
We'll have Herc position though, right?
Herc will be recovered before we deploy the next one.
Because you have to recover the mooring with Herc out of the
Yeah,
water.
we can't chase down the mooring with Herc in the water, so we release the mooring, it floats to the surface, then we take the 2 hours to recover Herc, then we go after the mooring, recover it, then we deploy the new one. That's just how we've always done it, yeah.
But the beacon on the, sorry, maybe I didn't hear or misunderstanding, but the beacon on the seafloor to mark the position of where we want it to land.
Yes.
But Herc will be down there before we release the mooring, right?
Yeah, exactly.
We could mark the position.
Yeah, so we can mark that
based on
position.
Herc.
Exactly, yeah. So we can use Herc to mark a position as a potential target. It's just a lot of times we like to have a beacon to aim for, so we know that there's nothing funny with the nav, and we're not, because it could be that 20 meters or something makes a difference.
Oh, we're planning to release the new mooring with Herc in the water.
No.
Pardon, I can't really hear you.
Are
What'd
we?
you say?
Oh, no. So how would we...
So Herc is going to release the mooring that is existing there.
Yeah.
So Herc will be there. We'll have a position from Herc.
Yeah, we
Oh,
will.
I see, yeah. So when you
Yeah.
put the new mooring in with a USBL, you just go off the old Herc ping
Yes.
from hours
Yeah.
earlier. Yeah.
So
okay.
that's what I mean with a backup plan. So if there
Yeah,
is
roger.
if there's enough scope so we can really move the cable out of the way and we've got a big landing area, then that's fine. We can do
Cool.
that. But if it's very narrow and we can't get the cable into a safe spot and we need to really nail it, then we'll put a beacon down first.
Got it.
Roger.
Because sometimes it's if you go down there and you mark a spot and then the current changes and these plumes kind of get in between you and your nav, that your nav tends to or sometimes can jump around a little bit.
Sure, sure.
But I also don't know. This year the nav seems a lot better than last year. Last year it had a big, depending on the ship's heading, it changed too, so I think it's been improved. I don't know what was done.
Yeah, there was an issue with the transducer last year, so that was sent in and repaired. The transceiver.
Yeah, so at least if you put a beacon down, you know it. You're exactly where you're going. I mean, there are 2 beacons down there. They're both, one of them's bound to work.
Yeah, it looks like both of these beacons were working on the previous deployment of them. So.
Yeah, they're both working on the node dive as well, so it should be okay.
So was the other one we put down this morning, though?
Pardon?
So the other one we put down this morning worked yesterday.
Which one did you put down this morning, sorry?
Wasn't it 2405?
Oh, I don't even, my mind's completely lost. This morning, we put one down where?
The autonomous southwest mooring?
Okay. That didn't have a beacon on it.
No, it did. It had 2405 on it.
Oh. Well, I didn't even know we're tracking that one.
Well, if the beacon doesn't work, we're not.
Where was the beacon?
Ooh, what's that, Fabio?
That was a
Oh yeah,
Salp,
yeah, you're right. Yeah,
Salp.
okay.
Cool.
Okay, there's definitely a beacon on that, and it would never work.
looked like.
It worked during our cable lay, but it didn't work
We
today.
didn't pick it up on the, oh, we never picked up the autonomous. I didn't hear that.
IG.
Well, sorry, I was completely confused there for a second.
So Fabio, what are you expecting on this to see on this transect, and what would be a real surprise?
Hmm. expecting to see a lot of gelatinous plankton, jellyfish, comb jellies. We're gonna definitely see a lot of myctophids, I suspect, from between 300 meters and 500 meters.
Oh, myctophids, better known as or more commonly known as lanternfish, my very most favorite.
Yeah. I'm not sure. There's always something new that we see. Like at the beginning of the dive, we saw a pretty cool Scyphozoan.
Scyphozoan?
Yeah. Look at that tiny little one at the left. That was a hyena jellyfish.
Cool. Sorry, the one we saw at the beginning, that I think I know is a fried egg jelly. Have you heard, is that the right term for that one?
What did you say?
Known as a fried egg jelly.
I'm not sure if that's the name.
Okay.
Yeah?
Locally we've got ones that have like a big yellow center and it looks like someone's cracked an egg in the water. So we call it a fried egg jelly.
Maybe that's that one. See, we started to see some fish now.
Yeah, there's a fishy.
At 200 meters.
Did you see that kind of slightly bigger fish at the beginning, Fabio? There was
it was kind of far though. I saw that was a interesting swimming.
Yeah,
definitely not common.
It did not look like a plankton fish. It looked like a fish that knew or had control over where it would go.
That's actually a great comment, Dirk, actually, that plankton is wanderer or drifter. So plankton includes anything that is kind of pushed or moved by the ocean current, including jellies. So it means that some of these fish, the really small ones, are also technically plankton. It depends on how big they are, and some of them go through various life stages where they're plankton and then they're not plankton. So yeah, it's a great observation that fish definitely looked like it was big enough to move itself around and thus is not a plankton. If I had my wish list, Fabio, I would be really excited if we came across some hatchet fish.
Ah.
Yeah, they're really, they're so ugly, they're cute. Yeah, they look like a hatchet. They look like the head of a hatchet.
Yeah, I know. When I had a freshwater aquarium, hatchet fish was a thing.
Yeah. There's freshwater hatchet fish as well. But the ones in the deep sea are creepy and ugly, but also so cute.
Yeah.
Yeah, I remember last summer during the Nautilus cruise, we had some interesting, not bycatch, but passengers on the ROV on the way up. One was a relatively big squid. Then we had that black, what was that black, remember that black eel looking thing that we had? With the expandable jaw, his jaw was...
eel?
Where? You saw that?
Yeah, we saw it got stuck on the ROV.
Oh, this previous dives here?
No, not this trip, last year. It was like a little black, long, slender.
I don't remember that one. I remember we got some myctophids that got stuck.
Yeah, we got the myctophids and...
I don't remember that one.
That's a beautiful jelly.
Wow.
What's that?
Here, I'll pull it up, actually.
It's
I've got a
a
picture.
ctenophore, actually.
A ctenophore.
And then we got the cool one this trip. What was that thing again?
That was a barracuda.
Yeah, barracuda.
Seems like
you like the way it sounds, eh?
Well, you kind of sound like you're going to say barracuda, and then it just kind of tapers off.
It comes, yeah. Dina means diminutive in Spanish.
So it's a little barracuda.
Little barracuda. Oh, cool.
Yeah.
Wide.
It did not look like your typical barracuda.
Yeah.
It looked more eel-like. Everything looks eel-like.
Yes.
But they have very cool purpley and bluish scales.
Let me see. I'm trying to...
Oh, sorry.
I took pictures, so I'll show you what that fish was.
I'm hoping you're about to see show us pictures of a gulper eel.
Gulper eel.
Yeah.
The thing, Monica, is that most of this pelagic fish, I mean, of course...
That's what it looked like.
Oh, that's a viper eel.
Yeah.
Yeah.
Oh, a viper eel.
We saw a viper. Look at the teeth on that
Oh,
thing.
yeah. Did we save that?
It came up with the ROV. It was just I don't know.
I think I saved that.
I think, you put it in a jar of some kind.
I think I brought it to the museum.
Then we saw these
Yeah,
with the little
those,
lights.
yeah. A lot of these fishes are disturbed by the propellers and the lights, and
I saw this one.
Yeah, it's kind of hard to bring fish in when you're making light and noise, and
Yeah.
Of course, they're not only does their vision tell them to get out of the way, but of course their
So this
the
one.
electric chemical or signature, pardon me, in the water tells them to get out of the way too.
So this guy. This
Yeah.
is like the vibrate.
I'm asking. I'm not telling. I'm asking.
So, sorry, you asked if the electric field?
Yeah, did the fish sense the ROV coming?
Oh, yeah, the vibration, the noise, the lights.
Yeah.
And, yeah, the turbulence. That's why it's very, I mean, doing ROV survey is not a very quantitative way to assess fish biomass or abundance.
But the squishies are less motivated to get out of the way?
Exactly.
Okay, cool.
The lack of brain may do it.
Lack of brain. Don't let things like that stop you.
The lack of a complex brain.
There you go. There you go. I think that's how I'm going to describe my next vacation. I'm going to go do something that doesn't require a complex brain. And just to go ahead.
Oh, that's another ctenophore there, it looks like.
More ctenophores, yeah.
Yeah.
Which ctenophores, if you really want to mess with someone, get them to spell that word. Starts with like a C-N-T.
No, C-T.
C-T, yeah.
N-E. No.
See? It's hard.
N-O-F-H-O-R-E. Here, on the screen.
C-T-E-N-O-P-H-O-R-E-S. Ctenophore. There you go.
Exactly.
And welcome to anybody who's just joining us as we are descending into the deep. This dive is taking us to part of the Endeavour vent field, and we're looking at a maximum depth of about 2,100 meters, give or take, depending on where we end up. And yeah, the purpose of the dive here is to look for a mooring that we're going to then release and reset. Which again, I just made that sound really simple. It's actually incredibly complicated, but that is the plan. And we're going through a nice slow descent to look for creatures, which we've had a few good cameos. There's a little myctophid that just showed up. Mytophid, sorry.
Were you just talking about the spelling of ctenophore?
Sure was.
Well, I got a joke
Hit me.
that I'm sure some people have heard before.
Mm.
Why couldn't the ctenophore hear anything in the ocean?
Ooh, why couldn't the ctenophore hear anything in the ocean, Trevor?
Because the sea is silent.
Because the sea is silent. Nice.
Oh, wow.
Ooh, you know, great dad joke on Father's Day. So shout out to any and all fathers out there. I see that today would be an excellent day for dad jokes. Now, Fabio, when you're reviewing this footage, does that do you do it more slowly, or do you do it at this pace, or do you do it frame by frame? Like, do you do more with it after?
Yeah, we actually do a lot of work frame by frame, and we initially do a lot of manual annotations. But maybe perhaps you're not here yesterday. We were talking about how we're applying artificial intelligence, machine learning model to go through all this. We had about 8,000 manual annotations of these jellyfish, comb jellies, and fish, etc.
Mm-hmm.
And now we train this model, and we're hoping to apply for all the 10 years that we have so far of this blue water transects to get the abundance and the biodiversity data from those transects. So it's in the works.
Wow.
Paulo Correa at ONC and a number of students are helping us with that. We also have support from MBARI, who has developed their own machine learning models, and we are gladly collaborating with them.
Super cool.
This project.
Ooh, look at that thing. It just went from kind of curvy to straight. Now's where I really need to access the tele thing, Majabber, so I could circle all the squigglies. What is that again? Telestrator, like Illustrator. Got it, Telestrator. There we go. Trevor, do you think you'll be doing any untangling today?
Nope.
Yep, I don't think so either.
I think not.
And folks are wondering what our greatest ocean jokes are. Anybody have another ocean joke they'd like to share? I'm seeing
Take a minute.
yeah, a few people have fear in their eyes. Don't make me tell a joke on the air. Quit that.
I don't think I have any other sea jokes in my head right now.
The only one I can think of is this one, and it's how do you make an octopus laugh? You give it tentacles. Yes, please hold your laughter. Yes.
There's one that's related to science, but not particularly about the ocean.
All right, go for it, Fabio.
What is the favorite? What scholars like the most for nuts? What kind of nuts scholars like? I'm trying to remember.
What kind of nuts do colors like?
Scholars.
Oh, what kind of nuts do scholars like? I don't know. What kind of nuts do scholars like, Fabio?
Academia nuts.
Oh, academia nuts. Oh, pardon me while I slap my knees.
You said that joke, so.
Yeah, pardon me as I
Here
slap
we
my
are.
knees. Whew. For those of you with
It should be academia, right?
Yep.
Yeah.
Not pronounced correctly.
Oh, you did great. It's awesome. I always love watching these little fish because they're kind of like, oh, I'll escape to the right. No, the left. No, the right. No, the left. No, the right. No, the left. And I can relate to that in terms of a decision-making process. Dirk, you were showing us some photos of some very unusual creatures, the eel and things like that. And as you said, we've seen a squid, and a couple of days ago we had another fish come up in the ROV, which is again relatively rare. But what's the coolest thing you've seen on a blue water dive, or kind of in your experience with Ocean Networks Canada and doing this stuff?
I don't know. I like. Can you hear? Is this working?
Yep, I can hear you great.
Okay. I'm not sure. Like today, for instance, it wasn't on the dive, but you know, we're 250 kilometers offshore, and there was a seal or like a sea lion next to the boat.
Oh yeah, that's
for
right.
Most of the afternoon. That's kind of cool.
Wow.
Yeah, I was checking this out.
Yeah, I mean, besides like just big halibut or weird rays and stuff that you wouldn't normally see because of how deep it is, I don't know. I've missed a lot of cool stuff while I was on cruises. Other people would see really interesting things, but
Oh, what's the one that haunts you?
oh, last year there's a whole pod of dolphins teasing the ROV as it was coming into the surface.
Oh,
yeah.
Yeah.
What about the
Missed it.
moonfish?
The moonfish.
Yeah, or you call it sunfish.
Oh, the sunfish.
Oh
no.
yes,
We
the
call
mola
it
mola.
We call it peixe lua in Brazil, so that's moon.
Yeah.
Cool.
I've never seen one on any ROV dive, but we see them from the vessel, from the boat.
I saw one at the very start of an ROV dive in like 2015, just as it hit the water, panned around and saw one.
Yeah.
And I went outside to see if I could see it, but of course it was nighttime. And then the second time we did actually get close enough to see a mola mola.
Oh.
We thought actually we had dropped a sediment trap, and our lead engineer at the time thought that it had floated, it had become buoyant. So we went to look at it because it was a big white thing, and we just dropped off a big white sediment trap, and turns out it was in fact a mola mola. So that was a highlight for me, and I'm a little greedy. I'd love to see another one because they just are so cool.
Yeah. What was it? A week ago we saw a whole, was it still a pod of sperm whales?
Yeah,
Swim
yeah.
by the boat. That was kind of cool.
Very cool.
Yeah.
Yeah, they were neat. Now, have anything come to mind for you?
Those are larvation
Sorry,
houses.
what's the question?
Mucus houses.
Oh,
Sorry.
larvation mucus houses. That's
Yeah.
not the question. Sorry. The question was, what's something cool that you've seen on these blue water dives?
Something cool in blue water.
Or perhaps you heard about that you missed?
Probably the coolest was a bunch of, are they oceanic white tip sharks? Is that a
Wow.
thing that exists?
Cool.
Yeah, that's cool.
And not just one, a bunch.
I
Light
forget
tips.
the name of them. I think that's right.
Okay, cool.
But there was a bunch when we were getting near the surface to recover the vehicles. They were kind of all swimming around and checking it out.
Neat.
That was cool.
That is awesome. Trevor, you look remarkably relaxed, but I am going to ask, what have you seen that's super cool?
I like being at the surface
Okay.
with the little birds on the surface
Oh, the
and
feet.
their little flapper feet. You
Oh,
tip the
yes.
Zeus up. You
Yeah,
see
that's
the
kinda
little
funny.
bird butts and their feet flopping around up there.
That is,
Oh my
that's
God,
pretty
that's
cute.
amazing.
It's a really cute perspective that you don't normally get to see.
I've never seen that.
Really?
I don't think so.
Yeah, when
Next time there's birds on the surface,
Yeah.
we'll be going
You see
back
it.
to Barkley. You'll see it there. Yeah.
That'd be awesome.
their little feet.
Oh, yes, I
Adorable.
would like to see that. I'm going to get up for that. But you just mentioned something really important there, Dirk, is that way out here, we don't see a lot of birds. We're way too far out for the birds.
Far out.
Yeah, far out. Partially because I think there's not as much upwelling, not as much food. Also probably not as many ships, so they're not as conditioned to come follow them. But yeah, we definitely see way more birds at Barkley, which is much closer into shore.
I saw
Yeah,
a bird
and
at Cascadia.
It's got the continental slope drop at Barkley Canyon there. So yeah, really productive for birds. We do get birds out here at Endeavour. I've seen at least 3, maybe 4 species since we've been out at Endeavour on this expedition.
What's
Yes,
the most
sorry, I shouldn't have-
unexpected species?
Shouldn't imply that they're not here completely, but they are definitely in much smaller numbers.
The diversity isn't as great out here.
Yeah, actually, Trevor, could you repeat that, please?
What's the most unexpected bird species you've seen out here?
I haven't seen anything unexpected yet. Everything has been fairly expected, although when we were transiting through Barkley the first time, there was a flock of gulls migrating out that direction, and I think they were Sabine gulls. So we don't usually see those around Vancouver Island, but they are known to migrate offshore towards the Arctic in this area.
Oh, neat.
Neat. I remember once being on a whale watch, and the guide confidently announced that if we wanted to just look to the left, there was a bunch of human skulls. And of course, everybody's like, excuse me? What he said was herring gulls. So no, it was just birds, herring gulls, which are very common.
We don't actually get a lot of herring gulls, at least in Victoria. They're considered uncommon most of the year and rare at other times of the year.
Well, maybe that's why he pointed them out. Yeah.
Oh, is that a
Oh,
squid?
nice squid just got by there.
Fabio, is a mola a plankton?
No,
No. No, thank you for asking. No.
it's a necton.
It's a nectin. Oh, tell us more. What's a nectin?
Well, necton are the larger organisms that have the ability to swim against the currents. The plankton, they just drift away, most part.
There you go. Now, the larva of mola or baby fish would be plankton for a while, yeah? Because they're so small. This is one of my favorite things about the mola is that the baby fish are like tiny, tiny,
Yeah.
tiny, tiny.
Exactly. They
Yeah.
even are called ichthyoplankton.
Ichthyoplankton.
Yes.
Cool.
Is that fish plankton?
Yeah.
Yeah.
Ichthyoplankton, yep.
Ichthyoplankton.
Bringing
So-
out your biology background there, Dirk.
Mhm.
Yeah, my crash course I took, I guess. Yeah.
Very good.
Well, I figured ichthyos is either birds or fish, so.
What is birds? Avia,
What are birds?
what are birds?
Yeah, I never
Birds
know.
are dinosaurs.
Dinosaurs. Yep. Fabio, you just became my new favorite person. Well done.
Well, they're definitely the most closely related.
Very good, very good. Lovely Gabby, would you like to tell us about the best thing you've seen?
She's all comms.
I am on comms. I was just thinking.
You're very, very faint. Are you sure you're on
Yeah,
comms?
the whole front row is kind
I'm
of quiet.
sure. Can you hear me now?
Yep,
Oh, yeah.
there you go, there you go.
I have seen crab-eater seals
Ooh,
hunting fish in the light of a camera sled. It's pretty cool. The camera sled like brought all the little fish and the krill around. That was pretty cool. A number of years ago now, maybe like close to 10 years ago, maybe, yeah, 10 years ago, Herc saw a sperm whale in the water column.
Yeah, that's
yeah.
that's a classic. Yeah,
That's
a good one.
definitely a classic worth checking out in the archives. Yeah. Yeah, it's fun to watch like the bigger critters come down to do their feeding right in front of the vehicle.
Yeah, I saw a seal hunting one time. It wasn't that deep. It was 70 meters or something. It was
Yeah.
pretty cool.
How deep
Someone
was
passed the
the
lights.
sperm
Yeah.
whale? Do you know, Gabby?
What's that?
How deep was the sperm whale scene?
I don't know. It could have been very deep because I think they were near the Puerto Rico Trench at the time. But yeah, that was in the Gulf of Mexico.
Neat. Ashley, what about you? And you are very quiet.
How about now?
Still pretty quiet, my friend.
Okay.
+6.
Can you hear me now?
Yep, go for it.
I haven't had any blue water surprises.
Oh, well, let us set the
Notable,
stage.
But the sperm whale keeps me engaged on descents for sure. The idea of one.
Very cool, very cool.
I think there was some sperm whales sighted off the ship on this cruise, wasn't there?
Yeah,
Yeah.
I
I heard
did
a
mention
rumor.
that. Yeah, about a week ago on our way, we saw 3 to 5 whales, and we
Yeah.
all kind of did our best to
I
identify
think
them.
it might be even more than
There
that,
was like
yeah.
a whole pod. There had to be over a dozen.
Ah,
Yeah.
maybe I just thought it was the same whale over and over again. I'm easy to fool like that.
You just forgot to look at the other side of the ship.
Right, yeah, that's just it.
There was
Oh,
another half dozen.
yeah,
on the
one majestic whale on one side and there's a dozen on the other side. That sounds like me. Yep. Maybe I think I've only ever seen one seagull in my life.
One seagull?
Yeah, they all look the same. So maybe I just kept assuming it was the same one.
There are a lot of them.
Even when you see 10 at once, it's all just the same one.
Yeah, exactly. Oh, my vision.
He wasn't on an ROV dive, but a subsea camera. Like Fabio's, maybe you saw some cool stuff on that camera at the node, Barkley node.
Oh yeah, where we had the fish acoustics
Yeah.
experiment. Yeah, we even saw a 6-gill shark
Yeah, very cool.
coming. We had a bait release system in that area in front of the camera. So yeah, we had quite a bit of scavengers attracted to the bait.
I've seen a 6-gill on a
The
ROV
interesting
dive.
thing that I
Oh,
thought,
squid.
there was one that,
Those are big, hey?
what's that, the squid?
No, the 6-gill.
Yeah, they're big,
Huge.
but what about the elephant seal? That was down at 600 meters on your camera.
Oh yeah, we're just publishing a paper actually about that.
Oh, spoiler, sorry.
You're spoiling it. It's,
Oh.
it's coming out
Okay,
this month.
redacted.
Well, we saw this cool behavior in summary of this. We identified 6 or 7 adult northern elephant seals. We have marks on their, bodies and stuff.
Yeah.
And they were coming to hunt sablefish, essentially. But they kind of take a nap. Oh man, I shouldn't be saying that. But
Oh, sorry. I'm spoiling this again.
Yeah.
Just
No,
wait for the
no,
paper. It's coming out
It's
soon.
coming out soon, but it's very kind of new in the sense that
Ooh, what's
What's
that?
that?
Whoa.
that's a Catino 4,
That's cool.
a special one. I forgot the name. I should bring it up. Anyway, so we also found that the seals are attracted to the Dietzen sonar because they
To the sonar. I was wondering
yeah.
how they get down there, because they can't smell subsea,
Yeah.
can they?
can smell. No, yeah, of course.
How do you
They
smell
have
without-
whiskers.
You can smell through a whisker.
Well, it's like the chemosensors, right? Or
Oh, wow. Okay, cool.
Cool.
So here's a question for you. How do elephant seals, if there's no Ditson, no ROV, no lights, and they're 600 meters deep, how do they hunt?
you're gonna read in the paper.
Oh, no. That's
Yeah.
how you get
There's a bunch of background.
the viewers up, huh? Just tease.
There's a tease.
I'm going to wait for
coming.
the movie.
Oh, it was
a
known.
blurb.
It was known that the elephant seals would dive deep, but they never observed like this repetitive behavior.
Yeah.
And of course, there's an attraction. The sablefish are first attracted to our infrastructure.
Yeah.
The lights, as an effect, they, as the lights turn on, they come close to the platform. They get agitated. And then the the elephant seals learned about that spot, and they came, and they will dive, and then you see them a few hours later. Well, they disappear, and then they come back on the next day at the same hour, and they take a nap. It was the funny part with you turn the lights on and the camera, and then they get, you know,
Well,
steered up, and they wake up, and then they start hunting.
oh, that's really cool.
No more information.
Well, some people are wondering, where will the paper be published? Where could they look for it?
When?
Where?
Where is the +1?
Plus
Journal.
one.
Yeah.
Okay,
coming
watch
out
for that. And I believe we also put at least a notification on our website as well, so that people are directed to them.
We'll have that information on
Yep.
it right now. Yeah.
Yeah. So once it's published, it'll be available on our website to direct you to it. So if you can't remember +1, then you can definitely head on over to Ocean Networks and check out at least some directional information. And yeah, really neat. I was going to mention as well, sorry, that I was going to say something and then it left my brain, but don't worry, it's back. Some folks might notice that when they go to the Ocean Networks Canada webpage, they will see live cameras, and they'll click on them and see that they're dark. And that's actually because of what Fabio was getting at. We want to reduce light pollution and reduce our interruption on the benthic environment. And so the lights come on at set intervals. And the interval is usually listed underneath the camera as well. So you can have a look and set your clock to that and check them out. But if you're ever on our page wondering, why is it dark? Well, it's dark because we're trying to look after the environment. You can also head on over to our C-Tube, and C-Tube will give you the recorded sections of video, so you can check those out as well. And Gabby, someone has written in and said that the sperm whale encounter was at approximately 600 meters. I'm getting a thumbs up, confirmed. And for us, I see that we are at about 500 meters, so if any sperm whales are in the area, you got 100 meters to get yourself over here. Chop, chop.
This isn't far from where we saw that pod the first day we arrived around here.
Oh, that is true. I wonder if they got my memo. My email, perhaps. Please show up at this time.
Your sea mail?
Oh, but
Sea whale.
sea whale. I do really like these little fish, though. They're pretty cool. And that must be another larvation? Or larvation house?
What is that? Oh, yeah.
Yeah, they make these mucus houses.
I'm just always so optimistic I'm going to see something really new, really cool. And
Sometimes you do.
it's so rare.
Sometimes you do.
Now, Fabio, all these little fish, they seem to be swimming downward. Is that, are they going with us, or is there anything about that, or is that just what they want to do?
They do vertical migration, the water column, so they actually, at night, they migrate upwards because they go after zooplankton. And during the day, a lot of zooplankton, they hide at depth, so they migrate downwards. So that's the largest migration on the planet at the scale of the entire ocean.
Cool.
I have a question, Fabio. What's the
Quick,
difference
quick.
between migrating and just moving?
Migrating and moving. Ooh,
Like, do humans migrate when they go to work in the morning?
I don't think so. Well, I think it's a directional, large-scale movement of a large population, or populations or communities.
That sounds like rush hour.
Rush hour, yeah.
Oh, this could be quite the battle of semantics.
It's a very philosophical question, or sociological, rather.
Oh, yeah. But obviously if these vertical migration, the fish...
Yeah, they
you know
come
what I'm
back
saying.
too, right?
They, if that counts as a migration, then I wonder how that's defined.
Maybe we can
Yeah.
flip it. Trevor, do you think that the migration counts as a commute?
I think so, absolutely. These fish are commuters.
Commuters, yeah.
Yeah,
The
absolutely.
biggest commute in the world. Happening right here, right now.
Right now, you're witnessing it. You're stuck in traffic? Don't feel bad. You're not stuck with the entire ocean.
Oh, it's all about perspective.
Yeah, I guess you should start using that terminology on your day-to-day
They're
basis.
just
You're
commuting.
just migrating to work.
Yeah.
Yeah, how was the drive in? Oh, it's migration. Man. I guess maybe it's different that we're not going to get picked off by predators. I mean, that would make the trip way more interesting.
I'd
What
hope
was
not.
that one? That's stickfish.
Every morning in rush hour, one person gets eaten by a bear. Somehow from within their car.
Well, the bear probably has a bigger car.
Yeah, I guess so.
Yeah. Is that actually why we see these squids, most likely? Is that they're preying on the fish?
Yeah, sometimes, yeah, the larger squids, they're really capable of chunking some of this tiny fish. Yeah. They have pretty powerful beaks.
That is true.
Fabio, what kind of fish is this, did you say?
These are myctophids, lanternfish. They're the most abundant fish in the ocean.
This one? These
This large group of myctophids.
These are the ones with the little lights. The
Yes.
cell clusters are cool.
Light organs and yeah.
Yeah. And this
So-
is true bioluminescence. They use luciferin and luciphase to make their light.
Yeah,
Do you think
very good.
do you think something like a sperm whale would be hunting by bioluminescence when they dive down?
It could
By
be.
tracking squid, following disturbed light from other animals. Like how?
Well, a
I
lot
just
of.
don't know how they would. How do you hunt something at a kilometer deep?
There's
at
a squid.
that.
Let's ask him.
Yeah.
That's a squid?
Yeah, look at him. Look
Yeah.
at his little head wings. Not a technical term.
Isn't it the Harry Potter owl?
Head wings? Oh, but I'm chill.
I think that's a good natus squid. Poralia.
I'm going to narrate for a second here. Fabio's got this fabulous table in front of him with all these different labels that he can use to ID things. And I'm glad that you know what you're looking for, because it looks like a bag of Skittles. There's all sorts of buttons in there.
That's a feature on our C-tube annotation interface. Is essentially a button set. So you create these buttons with the name of the species that you know that you're going to find in this region. So you kind of organize the button set. You know, this is Endeavor, this is the cluster of species I see in Endeavor, cluster that I see in Barclay Canyon. So it makes our life easier to... But this is just a first pass, right? After we go to the lab, we're going to look at the video in full and annotate much more carefully.
Are you annotating right now?
Yeah.
Cool.
It's a great question here about what does water column refer to? Is it the space above the vent? And water column is basically just referring to the shaft of water that we're in. And you can kind of think about it as like an elevator shaft where it goes to all the different floors. And so we basically kind of define an area in the ocean as the water column, just to kind of give it some parameters.
I missed the first part of your question, sorry.
Oh, just how would you...
Defining shaft.
water column. How would you define the water column?
Well, the water column is everything that's above the seabed.
Everything, yeah. So not just vents specifically, but basically from seabed to surface, we refer to as the water column.
Yes.
Yeah.
And of course, the water column, when we span a few kilometers in the ocean, you have several different regions.
Mm-hmm.
We have the epipelagic, the mesopelagic.
And those
the,
would be
the
like floors.
Well, yeah, kind of depth ranges. Like the bathypelagic, for example, is like if you go from 300 to 3,000 meters,
Mm-hmm.
and then you enter the abyssal pelagic, where you kind of in the depths of abyssal plains, but also in the water, let's say from 3,000 all the way to 6,000.
Mm-hmm.
And then you will enter adopelagic, where the trenches start
Oh yes, the Hadal
below
zone.
the Hadal zone,
Yes.
below 6 kilometers all the way to 11 kilometers.
Mm-hmm. So named for Hades.
So yeah, people give names to make things easier to study them, but it's just the blue water from the surface to the bottom of the ocean.
Cassie, my ping pong brain is ping ponging. What are some of the bird species you've seen so far? And the reason I'm asking you is you have been an amazing bird watcher. You've got your bird books, your binoculars, your camera. So that's why I'm calling you out, because you have been doing such a great job.
Well, I mean, there's only about 4 species for me to identify, so it's not a lot of detective work there. But yeah, some of the ones we've been seeing, you know, the common ones are the black-footed albatross. We see quite a few of those. And some of the little ones we're seeing are storm petrels. So far I've seen 2 species of those, the Leach's storm petrel and the fork-tailed storm petrel. As well as there's been a couple of different species of shearwater we've seen around. And then at Barkley, we also got to see some northern fulmars. That's about it of the more likely ones that we've seen out here.
Fabio, some of our viewers are wondering, the way you were describing the different zones and things like that, could that be analogous to the rainforest with different layers and zones in the rainforest?
Yeah, I think the stratum of the tree lines, right? But that's we're talking here a much smaller spatial scale, right? If you're talking about just 100 meters from the bottom of the soil to the top of the tallest tree. And here we're talking about thousands, a few kilometers, right?
Yeah, significantly more distance.
You can, in biology, you can, compartmentalize to make it easier for understanding and studying some ecosystems.
That's why we have phylum, because scientists love to phylum.
What? Well, yeah, that's the whole nomenclature, standard for giving names to species. Linnés, right? A few hundred years ago, they came up with this scheme of the Latin and the.
I'm going to interrupt you for a second. Is that a transparent tailed worm?
I think that's might be a swimmer. I don't know if that's that's a polychaete. No, maybe not. That's too big.
Yeah, I thought swimmers were like the little guys that are like really flappy.
Yeah, well, maybe that one is dead or just chilling. But yeah, that looks like a little siphonophore, maybe. I don't know.
We'll have to review the footage.
Yeah.
Instant replay.
Here's a question for you, Fabio. Any species that you thought might be edible and delicious? Now, I do know that the black cod and the sablefish are of commercial value, but is there anything else down here that is particularly tasty?
Oh, the squid you just saw.
Oh, that's right.
Oh, look at the shrimp.
Oh, yep, shrimp. Those are delicious.
Shrimp.
Poor thing, it's not his fault.
That's a cool shot.
Shrimp.
Shrimp.
I had to make sure that I actually logged that under shrimp and not tramp.
Tramp.
Yeah. 2 A's in tramp.
Hey, Cassie, a question for you.
Yes,
Looking
Fabio?
on C-tube here, dive in progress, there's a red tab, and it's called historical video.
Oh,
What does
yeah.
that mean?
That means you're not logging live, or are you logging live? Yeah.
Yeah.
You can like log in the past as well.
All right.
So sometimes when you like click on like an annotation you've already made,
Okay.
to like edit it, it'll like go into like the historical
Four
mode.
beams.
I don't know the specifics of how it all works.
I guess this is just a new feature because we do this all the time and I never seen this. Yeah. Okay.
Yeah, there's some new things with C-tube. It's been a little buggy with some of the stuff they rolled out before this expedition. So
Ooh, can we see the
yeah,
historical
that
altitude?
one might be
One of the new
yeah.
stuff.
A whale swam underneath
I
us.
have already a few recommendations for improvement.
Yeah, actually, if you could send those to us, that would be great. We take
Saw it just
feedback
now.
after all the expeditions so that we can make improvements before the next one.
Yeah.
Fabio, we were talking about the various layers. And how deep does something need to be considered abyssal? I think it's 3,000 meters, but it's also whether or not it comes off the continental slope, right? Because we're not quite 3,000 meters, but we are off the continental slope.
Yeah, we are at at the Cascada Basin. So a basin is in theory like a shallower kind of sea.
Mm-hmm.
So yeah, we're not properly at the Pacific abyssal plain yet.
Mm-hmm.
But I mean, they do consider relatively comparable in terms of the characteristics of the environment. Not here in Endeavour, of course, because we're in the ridge, but before when we were at Cascadia Basin at 2,600 meters.
Still considered abyssal?
Yeah.
Okay, cool. And some great questions in here about how many of us are currently in the van working. And there is, of course, myself chattering away. We also have our data logger. We have a dive leave. We have a scientist, navigation, 2 lovely and talented ROV pilots, and of course video. And at any time we could be joined by engineers, more scientists, other dive leads. Lots of room in here. But that's who we've got for now.
I'm currently the only one sitting on Hercules.
Currently only,
The only person sitting in Hercules. There's only room for one person in the vehicle.
Only one person. Yes, only room for one in the vehicle. No, Hercules is the only one in there. Yep. It's just himself, no people. It's like whenever you go for a medical test and they ask you to fill out, what's your relationship to the patient? I always write occupant. Yeah. I'm in here. Fabio, the interwebs want to know, have you ever eaten one of the specimens?
Well, one time we were in Antarctica, and maybe I shouldn't disclose this, but we were collecting sea pigs. We're trawling off the western Antarctic shelf and collecting a lot of sea cucumbers. We're analyzing their stomach contact, and so we're dissecting them. We had to dissect them inside the actual freezer, so it was a pretty daunting job. Then we saved a few sea pigs, and we decided to try how they they tasted. So we microwaved a few.
Microwaved? Of all the ways to prepare a sea pig, that would be my least first choice.
Eat it?
Yeah, we tried.
And?
Well, it tastes seawater.
Tastes like seawater. Alrighty.
Yeah,
There you go. Huh. Microwave sea pig. Go figure. I think if I had been there, which again I'd like to emphasize that I was not, I don't know that microwave would have been my first choice. I think I would have gone for like a nice, I don't know, maybe a nice bordelaise or a fricassee.
Let me see if there's another. Well, one time in New Zealand, I was on a ship, University of Wellington or actually NIWA, the National Oceanic and Atmospheric Institute there, and where they have a pretty big oceanographic vessel. They do a lot of fisheries studies. So they have a fish factory inside so they collect the fish and they already dissect them, separate the stomach to learn what the fish are eating. And we tried, so I don't know if you know about the orange roughy.
Yes, the orange roughy.
So the pretty long-lived fish, they're deep water fishes. They inhabited seamounts around New Zealand and Australia, and they're pretty much overfished.
Yeah.
So
yeah, sometimes called red snapper. Which
yeah, could be.
Yeah, because their other name, the Pacific slimehead, really not all that appetizing.
Yeah. Anyway, so I got the chance to eat some because they collected the fish for studying.
Right.
And they were serving on the ship because, of course, they don't want to let all that go to waste if you clean the fish and got the stomachs out and otoliths.
The otoliths allowing you to age the fish. So otoliths being the ear structure inside the fish, and like trees, the otolith will grow a ring for every year that the fish is alive. And unfortunately, like trees, once you kill them to count the rings, they don't grow anymore.
That's true.
Yeah.
You can take core samples of trees to figure out their age.
That is true. You can do that too.
Hard to do that with the inner ear of an animal.
Yeah, that does seem a little cruel. Yeah. But it's interesting that you mentioned that the fish didn't go to waste, because I know that there are some programs that go the other way with salmon, where commercial and recreational fishermen can donate the heads of their salmon to various organizations so they can dig out that otolith and check out how old the fish was at harvest and things like that. So that you get scientific data and food flowing both ways.
That's pretty interesting. Yeah, a few examples here in BC, right, about such programs.
Yeah, salmon
Sometimes
being
it's mandatory.
Pardon me? Go ahead.
Sometimes it's mandatory.
Yes, that's true. Yep. But it's great when the two systems can work together, because of course, we'd love to have lots of fish in the ocean so that anybody can fish them.
People who enjoy fishing enjoy good fish populations.
Yeah, exactly. I think Microwave Sea Pig is a great name for a band. I'm just trying to think of what music they would play. I feel like they'd be a hair band. Ooh, what's that? Oh, this is just really big fish.
What was the consistency of the sea pig?
Yeah, what was the consistency of Microwave Sea Pig?
It's pretty much filled with water, as it could, if you've seen a sea pig before.
Yeah,
you can see its guts from the body wall.
Right.
Because it's pretty much like, it's the gut full of mud, essentially, because they eat mud. And, it was pretty slimy. Yeah. Not that we ate more than like a square inch of that.
Right, I think you'd find that 1 or 2 bites is very definitive.
Oh, that reminds me of a very gross story I have.
Yes.
You are on SPL, I will warn you that, but after that, the choice is yours, my friend.
One time I was in a deep sea biology meeting in Iceland, Reykjavik, and they served us in one of the coffee breaks. They served us that rotten baskin shark that they bury.
Sturstrumming.
That's the name?
Yeah, well, I butchered it, but yes, sturstrumming fish. Yeah.
So that thing was the most, let's say, disgusting thing I've ever ate in my life.
Very strong ammonia flavor. I hear it's quite an acquired taste. I have not tried it myself, but yeah. Ammonia is not a delicious aroma. Interesting.
Did you say basking shark?
I think that's, or a Greenland shark, one of them, yeah. I think that's one of those that they buried for, I don't know, months to years, and then they let the ammonia cure the meat, and then they serve as delicatessen.
Well, you know, the reason the grocery store is so diverse is that somebody likes what's in there.
Yeah.
Question for the crew has doing this work changed your view on eating sea life? And what I'll add to that first. Whoa, look at that fish. That's cool. What I will add to that conversation, sorry, totally distracted here,
Wow,
is
that
the
looks like a pufferfish.
yeah, it did look like a pufferfish. That was really cool. Blah, 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 blah. Sorry, my train of thought left without any passengers. Where were we?
I don't know.
I don't listen to me either. It's all good. Ah, yes, eating sea life. So has done this work changed my views on eating sea life? And what I will say is that it's changed my view on making sure I know what I'm eating, because a lot of names for fish are culinary rather than biological, as we discussed, the orange roughy being called red snapper as opposed to a slimehead. So just being really aware of where that fish comes from. OceanWise has been a great resource for me on whether or not I'd like to consume or buy a certain fish product. And as Trevor mentioned, you know, having fish to fish is a big part of it. And OceanWise, their goal is that they don't want to be there. They want everybody to have all the fish they can choose. So I would say that's made me be more informed on my choices. But yeah, that's definitely something. Would anybody else like to comment on that subject? I won't name names in case you don't want to, but if you do, we'd love to hear your thoughts. Ooh, what is that?
I forgot the name of this fish. Actually, we have here in our marine field guide, I think. Just a sec.
Ooh, a
dragonfish.
Long-fin dragonfish.
Oh, cool.
That's cool. Long-fin dragonfish.
Oh, what's coming up here?
Some sort of...
Here we go. Look, it's eating.
Is that an octopus?
Something. Squid?
A squid?
No.
What is that?
Oh, we're gonna miss it. Dang.
Is that what we just saw in
I'm gonna have to
the
review it.
Argus cam?
Oh, and a viewer has corrected me. Thank you, viewer. Surströmming is Swedish fermented herring. Hakarl is the Icelandic fermented Greenland shark. So yes, thank you.
Oh, I got the shark right, at least.
Yeah.
Oh no, I got basking shark. They're similar.
Similar. Yes. You know, it's great. The
Oh,
internet
that's a tola.
has lots of information for us, so thank you there.
Some definitely different looking stuff here.
Monica, I do think the amount of time I spend underwater impacts what I eat.
Sorry, could you say that again a little bit louder? You're so quiet.
I was saying that I do think the amount of time I spend underwater impacts what I eat. I'm not a pescatarian, but I do have a list of fish that I won't eat again, and it always seems to grow.
Yeah.
Well, thank you for sharing that.
Oh.
And how we're going down to 1,000 meters for this survey?
Yeah, so 270 meters to go. Do you want to start counting, Trevor?
Oh, I'm counting.
In your head, right?
Yeah. 221.5 meters to go.
Dirk's also counting, I can see.
Me? No.
No, Dirk's not counting.
I'm thinking about how working with Ocean Networks Canada and being on these dives so often, every time we go to Barkley, we see hundreds, if not thousands, of those black cod, the sable fish,
Mm-hmm.
and everyone's always going off on how good they are to eat and how buttery the meat is. So last time I saw that at the grocery store, I was like, I gotta buy some black cod.
And how
It's
did
very
you find
expensive,
it?
right?
It's very expensive, and it was a very weird taste for me. I don't know.
Yeah,
I feel like
it's not my favorite too.
I feel like it was super buttery, but a very strange texture. I think it would
Yeah.
make probably good fish and chips,
Maybe.
Yeah.
but it was unusual. I like salmon. I think
Yep.
it's probably my favorite.
Well, again, like I said, the reason the grocery store is full of so many different choices is somebody likes that. So yeah, someone likes it.
I can see why someone would like it.
Yeah, fair.
That's very unique.
But yes, I've had friends have it and say that it has a very buttery but almost fatty texture, so it is different than what you're expecting with a normal fish.
Yeah, it's strange.
Got an interesting question from a viewer in New Zealand, wondering how many squid there are on Earth. And I definitely don't have an answer for that, but one thing I do find fascinating is if you think about how much squid you'd need to support a sperm whale, which we know that that's a fundamental part of their diet, and you assume that 10% of the mass goes up through the food chain, you need like, I don't know, 300 tons of squid to support the sperm whale population? Like lots. How many squid do you think there are, Fabio?
Oh, it's gotta be tons.
I won't guess. I won't make a guess.
No, that's
A lot.
Price is Right rules?
On the zillions scale.
Zillion scale. There you go.
Isn't
Zillion scale.
squids everywhere, right? Like
Yeah,
it's
very
in
common.
every.
Yeah, very common.
Yeah.
And very different life plans. Like some of them live several years, some of them are seasonal only.
Life plans.
Life. Well, yeah, I did make
Some
it sound like they
commute,
want to go
some
to. Yeah, did make it sound like some of them want to go to college, have a family. Yeah, you're right. What I meant was some live for a year or so, and some live multi-years. But maybe
There's
some
at
of them.
least 3.
Yeah. And Dirk, some viewers wondering for an update on the cable lay. How did that go? Can you give us an update on the cable lay?
I think in a nutshell, the cable lay went as expected. Yeah, I think successful.
Excellent.
We did test the cable after it was laid, and it worked. So that it wasn't compromised or, I guess, destroyed during the lay. So it kind of ended up how we expected it would.
Excellent.
Meets expectations.
Hey, that's good. Meets expectations, right?
Yeah.
Protractor.
Oh, there's a squid.
Yeah.
Oh, it's a fish.
Oh, yeah.
Wow, he does a great squid impression. Well done.
Hide as a squid. That's not the right strategy.
Oh, well, maybe. I'm reminded of, again, Finding Nemo, where they turn into the bigger fish. They turn into the Sydney Opera House, the big school. They're like,
Oh, yeah.
look what we can make. Yep.
But I feel like squid's bait too.
Maybe if they're a big enough squid?
Is the protractor.
I don't know.
Whee.
I also get the sense that Fabio is going to be very carefully checking my annotations.
Height measure.
Your annotations?
Yeah, I identified as a fish as a squid. That's not off to a good start.
What am I looking at? Oh yeah, height is 8.844 meters. Cool. That's how tall it is. Nice. How do you unheight? Oh no. That's the measurator. That's super useful, actually. That's the measuring tape.
Trevor, you're on SPL. What are you doing?
Measuring.
Ooh, what you measuring?
Measuring. Sonar targets.
Sonar targets. Very cool. Is that triangulating?
It's whatever shape you want,
Ooh.
as long as it's a line.
Any shape you want, as long as it's a line.
I don't know how to not show height anymore, though. Hide. Height. Unhide. Unshow. Cursor. Cursory. Ooh, the height of the height box. Oh my goodness. Ooh, here we go. Yeah. Whee.
I thought
Okay.
you said you could only do lines, Trevor.
Yeah, well, for the measurator. This isn't a measurator.
Oh, that's just drawn.
This is an indicator
Oh.
of the height.
Can we use it to play Pictionary?
I don't think so.
No.
I can wiggle it around a lot, and that's about all I got.
Oh, that's cool too.
That is a cool feature that I didn't know was on the sonar.
That you could wiggle it around a lot?
Yeah.
Yeah.
No, the measurator, not the
measurator's
box.
good. Yeah,
the
the box
box,
isn't
I don't get much from the box.
I don't know. The box isn't good. Oh, I'm in a target mode now, and I can't untarget.
It comes at a price.
Yeah. What if I click over there? Okay, good enough. What's this? Scan. Oh no. Control head. Display settings. Targets. Measurationals. T-square. Projector. Text.
I'm excited to get to the bottom on this dive, though.
Wiper.
Yes,
I
definitely.
think
Ooh,
in my 8 years at ONC,
Wipe.
I've only ever...
Yes.
I
Wipe.
think I've only ever seen this area once or twice.
Wipe
It's
this
rare
one.
that we go here.
that is true. We
Wipe.
don't go here very often, and it
Good.
is a really unique place.
That's text.
Yeah.
Text
Very
only.
similar to Main Endeavour Field, except the I guess the black smokers we see here are more
That's
very
text.
tall, narrow,
Is it text
skinny
only?
pillar-like spires. They're about 30 meters tall. So
Cool.
yeah,
Very cool.
we'll have a quick look, and then we're going to start the dive with mapping the
Ooh.
kind of the immediate area with multi-beam,
Ooh.
and then after that we'll
Would that be the orbit?
W with an orbit, yeah.
There we go.
Very
And
good.
then we'll take some gas tight samples from the vents themselves,
Yeah.
followed by
Gauges
some
and the ground
photogrammetry
faults.
where we build 3D
They're all
models
good.
of
They're all good.
the spires by stitching photos.
Yeah. The photogrammetry is great because you get that real long look.
Yeah, that's really cool.
Oh,
The,
yeah.
the product that comes from it is amazing.
Absolutely. We a few years ago there was one that was made and displayed in a few museums as well. But I think you can use it for 3D printing too.
Oh, yeah, I think it's these models built from the photogrammetry. You can use it for measuring and doing stuff like that off of it, right, Fabio?
Oh yeah, so the idea is to, if you reconstruct the 3D imagery from these vents and these vertical structures, you can create a time lapse of this if you come multiple years and see how the chimneys are growing or collapsing. We come here this year after a sequence of massive earthquake activity earlier in March, and so we're very curious to see how these three-dimensional structures have changed.
Yeah. Especially if we keep seeing kind of that activity of the increasing earthquake activity, like there's been some trend, maybe coming back here more regularly and doing photogrammetry and multi-beam and seeing how it's changing.
We're not doing this dive though, right?
This dive.
This dive?
So yeah, the plan now is this dive, and if we don't get to it, we'll come back to it. I'm
Okay.
pretty sure it's in here.
It's not on the right objectives here, but
Now, will this be?
Yeah, the first thing after we hit bottom is
Oh,
to
okay.
survey with the the
Multi-beam,
acoustic
yeah.
multi-beam.
Yeah,
So will that be high res or low res mapping?
it depends what you consider high res or low res. Right now we have multi-beam of the area, but I think it was, I don't know what the kind of the box size is. I think it's like a meter size. This will be much smaller than that.
Cool.
Smaller than 10 centimeters anyways, is what we'll get from the multi-beam. The photogrammetry is much more detail.
More detailed than 10 centimeters?
Oh yeah,
Wow.
millimeter scale, I'd say.
yeah.
some.
No, the product from the photogrammetry is amazing.
Very cool. And how long is this dive intended to be?
So basically we have this area is called
And how many A's is in that?
high rise.
High rise.
Because yeah, I just know it as Godzilla because that's like the one that we go to. But sorry, what was your question? That completely threw me off.
Oh, how long the dive is intended to be?
All right, so it's a two-part dive. So the first part we're gonna do, it's more gonna be very much science-y and looking at things. So that will last till about 3:30, and at that point we have to transit over to the RC mooring site. That's gonna be about an hour transit, and then that's when we'll do our CTD calibration, not calibrations, but checks on the CT along the mooring. And we hope to release that mooring to the surface at first light. So about just before 7 o'clock in the morning, we'll release it.
I'm familiar with that time.
Yeah?
Yeah, 4 to 8 shift. I've become very familiar with that time, but go on.
All right, okay. And then I know that time is breakfast.
Mm-hmm.
But yeah, so then we'll release the mooring. So, and then after that, we'll just recover the ROV. So I would say the sciencey part of this dive is going to last till about 3:30. After that, it's going to be more kind of engineering. I don't know. It's not really engineering, but more like
Boring, is it?
more like interacting with infrastructure rather than interacting with science, with biology or
it's all
geology or whatever that is. Yeah.
Right. It's an educational salad. We need a little of everything.
Yeah. Yeah. So the first, that's why I like these vertical transects because they're only at the top kilometer.
Right.
But you get the scientists in here and they're kind of invested. After that, it's just winch goes down, hit the bottom, and then on the sciencey dives, it's nice because there's a lot of skin in the game. So you get to learn more about what's actually going on when the scientist is here, right? And then the rest of the stuff, like what my department deals with, is more the infrastructure. So that's what's the cable, what's the voltage, what's the junction boxes, what are they doing? Like what's the status of an instrument and swapping them and maintaining that kind of infrastructure.
Yeah.
So that's
cleaning,
you know, replacing parts, laying cable, that's more us. So the science part's more interesting.
Well, as the person who gets to ask what you're doing all the time, I think they're both pretty interesting.
Yeah.
Yeah, I think that's what makes Ocean Networks Canada a pretty interesting project, right? There's a lot of engineering, a lot of science.
Yeah.
A lot of education.
Yep.
And outreach as well.
All of that. A couple of wrapping up questions here before we kick it into high gear. Someone is asking if there's places in the ocean that don't have squids. Do all the oceans have squids, other than maybe Antarctica?
I would say I don't know. I think they are everywhere, but when we reach the ADO zones, you have less food, and yeah, maybe just a giant squid. I don't know. It's not a question. I wouldn't know how to answer that.
No, fair enough.
I'm sure they are everywhere in the ocean, but
they do seem like quite a ubiquitous species. Like they do seem to be kind of adapted to.
Well, we're talking about a lot of species, not one. Squids are
right.
Squids is a group of. There's lots of species.
Yeah. And someone's wondering if we've ever seen a colossal squid, and do they exist? I definitely think they exist, and I've seen one in a museum. But have you ever seen a living one?
A living one, no.
No, not yet. Though, yeah. Hey, maybe today's the day. I know Video said they had a good feeling about it, so hey, let's find somewhere between holding our breath and not holding our breath.
Good idea.
Yep. And multi-beam, that's a tiny little LiDAR, is that correct? Dirk?
Oh. Yeah.
Oh, your mic broke up there for a second. Thank you for that detailed explanation.
Is this thing on? Yeah.
Yeah, I hope you got that. Yeah, ooh. Thanks, that was too detailed to ever go back and say again.
Yeah. No, I'm not 100% sure.
Science magic.
Science magic.
Okay, we'll take it. Yeah.
I think physics magic.
Maybe this is one Nav can answer, if Nav isn't too busy.
Nav? Do you know what? Can you help us with multi-beam? What is that? Is that LiDAR? Is that something else?
Multibeam is a sonar, so it's an
Ooh.
acoustic instrument. And we're essentially sending a sound pulse out into the ocean and listening for the echo off the seafloor.
Mm-hmm.
We know the speed of sound in water, so
Mm-hmm.
and we know very carefully what time we sent the sound out and what time we received the echo. And so we can calculate distances based on that and know what the depth of the seafloor is.
Very cool. And is it true you can also get maybe a hint of what the seafloor is made out of?
Sometimes. So we can look at the backscatter value, which is essentially the strength of the echo, how loud it is, the intensity of the echo. And for, you know, like hard substrates like rock, the intensity tends to be higher, and for soft substrates like mud, it tends to be lower.
Okay, cool. So a little art within the science.
Yeah.
Cool. It's also what I like to sprinkle in there sometimes. I see we've lost all of our mytophid fish. Or they lost us.
Yeah, they don't passing like almost 1,000 meters. They're much less abundant here. Well, they actually don't occur all that deep.
I think we're coming up on a dinner plate jelly.
Yeah.
They always remind me of like an eye with eyelashes. Like ocean eye looking at ya.
That's a cool looking one.
Yeah.
So one other scientific objective of looking at this time series of pelagic transects is to look at the effects of marine heat waves. We have had quite a few marine heat waves here hitting the Northeast Pacific. If you remember the warm blob between
Yes, I do. Yep.
2013 and 16, kind of 15, 16, where the pelagic environment completely shifted the species that were occurring here. There were warm water species migrated from south and invaded this area. Lots of pyrosomes.
Oh yes,
It
all the
was,
pyrosomes.
yeah,
Hundreds of them, or thousands.
yeah, and it switched. The whole community shifted. Even zooplankton and phytoplankton, they completely changed. And the
Was
food
that a
web.
permanent?
No, that was like,
just the one summer.
no, not one. That was like quite a couple years actually.
Oh, wow.
Gulf warmer. The temperatures were 3 degrees above, in average, above the 30-year mean,
Well,
previous mean. And we had another marine heat wave in 1920, when we had all those crazy fires as well in BC.
oh, 2020.
Yeah, 29,
Oh yeah,
2019.
2019. I was like, oh, 1920,
Yeah.
that's ages ago.
2020. Yeah, we seem to have been having more frequent marine heat waves in this area, so we're kind of wanting to see what those changes, how they affect the plankton and the pelagic environment.
Do you find, with all the smoke and haze that we've been seeing from the fires, does that change anything in the surface of the ocean? You know what I kind of know what I'm saying? Like, does that add any nutrients that weren't there before, or is there any kind of research into that
Well,
that you know of?
there's definitely dust that settles in the ocean that is a major source of iron.
Yeah.
So actually we are in an area of the Pacific where it's like high nutrient, but well, there's some micronutrients that need to be fed from rivers or dusts from atmospheric dust. I'm not sure if there's been some research done specifically to address the fire, the smoke, and how that affects nutrient input into the ocean.
Like,
but
Victoria itself doesn't get much fires, but we see the interior fires come over it, and it's just hazy. The whole area is just hazy. I wonder if that can actually add nutrients to that surface.
I know there was a study in Australia where they looked at the effect of the bushfires in Australia, and that caused, and again, I'm quoting it in very simple terms, but essentially that caused a dump of nutrients via the ash into the ocean, and then that caused a plankton bloom in the Southern Ocean, which actually offset the carbon that was created during the bushfire. And so they just sort of canceled each other out very brief. And again, I'm very much, very much over summarizing, but I do know that someone did look at the effect of the bushfires in Australia. So I don't know if there's a study here in BC, but I do know that there was one in Australia.
Yeah, I'm just thinking because it's
1,000 meters.
There gets so thick you can sometimes.
I'm gonna speed up.
It's definitely very visible for sure.
It's time for full beans.
7 meters to go.
Yeah, not yet. 7 meters.
7 meters. Why?
You're looking at Argus, my friend. They're looking at Herc.
Yeah,
we're
looking
about Argus. Yeah.
Well, it's over, Fabio.
What? I'm looking at Argus.
You've been looking at Atalanta
the whole time?
Sorry, I called it Argus, but that's Atalanta. My apologies.
Sorry? Yeah, I hit the gas pedal.
Antsy.
Okay, we can log the end of the transect, Cassie. If you're already hasn't.
Will do.
Thank you very much. And
There's a great question here, actually, about the tidal action. Do you see differences in tidal action at this location?
Yes,
Please tell us more about that.
Well, even at the hydrothermal vents, the organisms there, that we have cameras, right, looking at this hydrothermal, the tube worms and the benthic communities, and yeah, we find the pressure, hydrostatic pressure from the tidal signals. They kind of affect how much sulfide is being released from the seafloor.
Interesting.
So that affects how the species migrate in and out of the tube worm bush, let's say. So it's kind of a flowing when you have high tides and high pressure so you have less release of sulfide from the seafloor. So then species, it becomes a bit less toxic, right? Sulfide is a bit toxic for some species. And then when there's low tide, these animals kind of get out of the tube worm bush to the surface.
Cool.
So yeah, at 2 and 2.5 kilometers, yeah, pretty much they still sense the tides, and yeah.
Very interesting. Do they also seem to respond to night and day? Are there any patterns that are diurnal?
Not in here. I think we're too deep. At least the species that we're studying here, but in Barkley Canyon, yes, we do dab some diel at the slope. Okay. We have some patterns that are somewhat tied to the day-night cycles.
Interesting. Thank you.
You're welcome.
Pardon? Yeah. No, I think these smokers are going to be really cool. The only time that I've seen is when we were looking for that bars, that autonomous bars that was engulfed by the smoker, and we couldn't recover it. It was part of the rock at that point. At this site, yeah, it's very active.
Can you tell us a little bit more about that, Dirk? Kind of start at the top. What are we looking for and what happened to it?
So a few years ago, one of our instruments, it's a benthic and what is it?
Resistivity sensor?
Yeah, and a resistivity sensor.
A bars.
A bars, yeah.
Yep.
And we installed it in one of the vents, but these vents are constantly growing, and they actually grow quite fast. And so it was an autonomous instrument because we don't have cables going through this high-rise area.
Mm-hmm.
And when we came to look for it, and because this area is so active with the venting and all the stuff, all the different changes in temperature and stuff, the navigation's not very good. So we can't really use our multi-beam system as effectively, or not multi-beam, our USBL system, which is how we track where the ROV is.
Mm-hmm.
It's offset depending on the tides, depending on the ship orientation, stuff like that. So we couldn't actually find it the first year we went looking for it, when the instrument had run and collected its data after the first year. So we came back 2 years after that to come and look for it again, and at that point we found it, because we looked more and we found it finally.
Better data.
The rock had completely, the spire had completely grown in around our instrument.
Oh.
And so you could still see a piece of it.
So you knew where it was?
You could see it, but you couldn't fish it out of the rock. The rock had kind of engulfed it.
Oh, interesting. What are the chances that it will recover it this time?
0.
0.
No, you can't see it anymore. It'll be gone. You wouldn't be able to tell. It'll just be rock all around.
Okay.
And when I say rock, it's like sulfide crystallization, right? So
yeah, it's rock.
Rock, mineral, rock, yeah.
Yeah, stuff.
Stuff, yeah.
Yeah, when in doubt, stuff.
Yeah, that's kind of cool, and that's the only ever time we were only there passing through, so it was about a couple hours we tried, and then off we went.
Very good. Alrighty, well, we're just doing a crew change here, folks, and I will say goodnight, so good night all. Thank you for listening, and I will hand it over to the lovely Brandon. All right, he's giving us a wave. He's ready to go.
No, we'll dive, we'll move on from high rise at about 3:30. Yeah, the ROV will be recovered. No. Let me finish my sentence. We'll recover the ROV probably around 8. It'll be on surface, 8 tomorrow morning.
Hello, everyone.
Hello. Rennie.
Good evening.
Good evening. On the descent, why don't I mess around with setting up an orbit?
Yeah.
Is the Norbit powered on?
Let me check my 17
Yes. Check them all.
No, yeah, Norbert is on.
Okay, cool. Great.
Oh, look at that temp probe go. 333. That's what we like to see.
Temperature agreeance. I'm gonna pay less attention to nav world while I do this.
So everything's normal. I'm gonna peek over your shoulder to see if I can remember Ross Prague.
I'm
Oh yeah,
I've got this thing in front of me. I'm gonna make sure I don't miss a step.
well, I got to this point before, and all I saw was like 4 start buttons, and I got nervous.
That's understandable. I get it. That's why I got this thing.
So I just KVM'd out of there.
Norbit's on. It's on. KVM'd out of. All right.
It's a good term.
Yeah.
KVM'd. One of the steps, if you run into some troubleshooting, there's like a couple of ways to get it through, and then like A, B, and then letter C is panic and desperately search for the nearest data engineer.
Is that as written?
Yep.
Panic and
Is that you?
there was no fidget spinner. What am I supposed to do with my hands?
Lynette's bringing fidget spinners up.
Yeah?
Yeah, she's about to bring some fidget cubes and stuff.
Oh, I like the cubes.
Yeah.
With the little buttons.
So she bought them last year. She bought so many.
This is like such a non-fidget world that we're in right now. Like it's all built in.
There's so many fidgets.
I do like these though.
Yeah, those are good. That one's a really cool one that Trevor has. Careful, it falls apart though. Then you gotta put it back together. That's part of the fidgeting. Yeah, you'll find out.
Ooh.
Let's see. We'll call this.
A little delta action going on.
If I don't watch it, it slides up.
Oh,
Push
yeah.
it back down and it slides back up. I might switch this around. Sometimes that works better. Precision maneuver here. Let's try that on for size.
What's your preference in the setup, Trevor? Do you like the stop at 50, get all set up, turn around, and or just do it all down there?
This is why we can't have too many fidget spinners.
Yeah.
We got too many things. Rennie,
did Steve give you an idea of what the exact areas we want to map?
Slow down.
I can show you on HiPAC what I was thinking.
Okay, sweet.
Are you looking at HiPAC?
No, let me look at HiPAC 1 second.
Okay.
Oh, you're into the boot.
HiPAC plan, HiPAC survey?
Survey.
All right, yeah.
So, Godzilla and Bambi, Boardwalk, Park Place, this whole high rise.
Yeah, okay.
So my thought was we'll do a pass kind of close by, but in this direction. Looks like 175 meters.
Yeah.
Go over, and we'll do another one down.
Yeah.
So that should cover a lot of it, but some of the tall vents like Godzilla, I'm thinking like we can do these two passes at 30 meters off the deck, and then we'll do I kind of want to see about doing one right down the center, but it'll be higher up.
Yeah, that sounds cool.
And then I think that ought to do it. I don't know how much, without getting super into the processing, I don't know how much, we'll have what we have basically, like whatever plume water mass difficulties we have with interfering with the acoustics are kind of going to be baked in, and there's not much we will be able to avoid that. And I think that this pattern allows for at least a decent amount of capability.
Okay.
And I would presume that that's covering the whole area, so I'd presume that that's what Steve would be fine with.
Yeah, I think if you go like that little
it
piece
doesn't take
just
long
below
to
Baltic,
go.
you know?
get all that. This whole kind of outcrop of spires would be nice.
I think that ought to cover. I don't think it'll take that long to do a few 150-meter moves.
Yeah, no, perfect. That sounds good.
Yeah, and we might want to try to establish the height of Godzilla so we know when we do that over the vent pass, we, well, first of all, just to orient ourselves and make sure our map here is as bang on as we want, or as we can get, and just to get a height for that final pass over the vents.
Yeah, okay.
Yeah, we'll just kind of see as we go, coverage-wise, and if we need one that kind of like a cross line to try to lock things together, we can try for that. I know we don't want to like spend a massive amount of time, but
yeah. So, are you comfortable with how the dive plan is written in terms of first finding these vents, kind of putting a real mark on where they are, check our nav, see how tall they are, and then starting the survey?
That sounds good. I mean, for the sake of time, I don't know that we need to visit all of them.
No, I think let's visit
that one. Focus on Godzilla.
Yeah, exactly.
That sounds good to me.
And are we pulling temperature and gas samples from any of the vents?
Yeah, we're going to pick 2 spots. So Steve, who's the scientist, that's kind of his focus, are the hydrothermal vents. And he'll be joining us probably soon. He's just sleeping right now because this is like his dive, right? He's passionate about this stuff. So once we hit bottom, he'll come down and he can walk us through why certain vents and why we're doing that. But we have 2 gas tight samplers, which are the vent plume samplers on the ROV right now. So he just kind of has to pick a spot.
Great.
But for anyone who hasn't seen that, that's kind of an interesting process. You survey a few of the spires, you look at a few of the spots, use the temperature. You first ream the spire or the hole where the smoke or minerals are coming out of. You take a temperature probe and you measure the temperature, and if it's a suitable, I guess if the parameters are suitable, you take a gas light sample from that spot.
Yeah, perfect.
But it's kind of cool.
Yeah, I mean, having only, this is the return for us here, and so I've only seen this once. That temperature gradient is what I was most blown away by. That's just like cold ocean water.
It's like 1 degree water, and then jumps up to 330 on our last dive, or 338. That's pretty impressive.
Driving like me there.
That's Celsius too. I don't know what that is in Fahrenheit for the US.
650, something like
Heading's all fun.
Yeah, it's impressive. Yeah, I mean, if cable laying wasn't your thing, maybe this is your thing. Hydrothermal vents might be your...
Cable laying was everybody's thing. That was a thrill ride from start to finish.
Yeah, futzing around with tangled ropes and
counting
mud
loops.
mats and counting loops and surveying a long stretch of mud.
Yeah, I feel that.
That didn't get you excited. Hydrothermal vents.
Yeah, someone has to get the short straw and look at these amazing geological features.
Like, I mean, this is a bit of a crowd favorite, right? Everyone likes the vents. It's because that's kind of...
Because they're awesome.
It doesn't come... Yeah, but I mean, don't forget Barkley. Barkley is also really cool.
Where?
What's cool about Barkley?
Barkley is the amount of life. That's the crazy part about Barkley. Like, just so many different kinds of fish and big schools of, like... I like it because it's, like... sizable fish. You're getting big sable fish. If you go Barkley Upper Slope, which we will be visiting as well, you'll see halibut, all kinds of stuff. Dogfish. Yeah. So we'll spend some time there. There's like a little bit of like a quick check that we're doing, and what that is we're deploying a hydrophone at Barkley Upper Slope, which is 400 meters deep, and there's a sound channel that sound kind of gets trapped in the ocean, and it travels along that channel, reflecting off of the whatever. It's not really, I don't fully understand it, but what the scientist is looking at is there's a sound source in Hawaii, right in the sound channel, and then it travels all over in that channel. And we're going to see if we can pick it up at Barkley Upper Slope. So we're going to